All right, good afternoon. My name is Rosa Cromrtie. I will be giving my poster on the application of screen printed electrodes for the detection of opioids in sweat. So as we know, there's a multitude of opioid overdoses and overdose related deaths where there needs to be a easy, rapid to use, and portable biosensing technology that is sensitive and can simultaneously detect opioids in biological fluids. So since there's little research available for portable drug detection biosensing applications, my aim for this project has been to develop a screening method using applications of electrochemistry and active sensing to detect the presence of opioids in sweat. Here you can see the materials that were used for this project. We used the modified aptamer, where you can see the sequence. 50, a gold nanoparticle that was in size 50 nanometers, in size, conductive carbon and silver paste to create the electrode, and chromatographic paper in a wax printer. So the processed material that we use can be seen in figure 1 for the electrode. We use the screen printing machine to create the electrode, and then we put the carbon paste and the silver paste onto the screen printing machine that is placed so that we can make the electrodes on top of the wax printed design. And then we use the potentiometer to measure all the voltammetric measurements that you will see in this presentation. In figure 2, we have a schematic design of how my target binds to the working electrode for the screen printed electrode. First, we have nanoparticles that were drop casted. The nanoparticles are the yellow that you can see, and the working electrode is the silver. Silver is supposed to be like carbon. It's supposed to come out black, but it's gray. Which is the working electrode. We allowed the gold nanoparticle to immobilize onto the carbon surface. And so after that, we followed by adding the thiolated aptamer onto that. So the thiolated aptamer can bind to the gold nanoparticle. And then in the presence of coating inside of potassium ferrocyanide, we were able to electrochemically analyze the presence of coating. So here you can see that in part C of the schematic design, you can see that there's a switch-on assay that occurs. So after binding with coating analyte, the aptamer structure takes on a conformational change. This shows that during the electrochemical sensing process that occurs, the oxidation of ferrocyanide and causes by the oxidation of ferrocyanide. So when the aptamer folds over after collecting coating, it begins to block the ferrocyanide from accessing the electrode surface. So it inhibits the ferrocyanide to create a electron transfer it blocks it from creating an electron transfer between the electrode surface and the ferrocyanide. So the aptamer blocks. It's the blocking mechanism for it. And in the table that you see below for under characterization, we were able to characterize the gold nanoparticles by size and charge, comparing bare gold nanoparticles with aptamer-coated gold nanoparticles. And it can be seen that the size of the aptamer-coated gold nanoparticle increases when compared to the bare gold nanoparticle. This change in size demonstrates the addition of DNA because we're able to see that DNA became bound to the gold nanoparticle because of the strength between gold and sulfur from the thiolated aptamer and creating a high binding energy. So we're also able to see that the charge of the aptamer coated gold nanoparticle shows an increase in charge, which is shown under zeta potential. Because the aptamer is blocking the surface of the charge of the gold nanoparticles to the electrostatic potential. So as I showed you in the previous schematic design, the aptamer will block the ferrocyanide from creating electron transfer between the surface. So that this coincides with what we see in part C. And in figure 3, we have scanning electrode micrograms to show the interaction between gold nanoparticles and aptamers at a size of 1 micron. The right image shows that when aptamers are functionalized on the gold nanoparticles, the size of the gold nanoparticles begin to increase, creating an enhanced cluster. As you can see, like for the left, you see the left is smaller, and then for the right is really big clusters of the gold nanoparticles that's being shown. And in figure 4, we have a cyclic voltammogram of various concentrations in coating, which shows where we can see that there is a signal drop as coating increases. So the more coating that is present, it blocks the reaction from binding with the ferrocyanide, which goes back to figure 2. Figure 2 for the end of the schematic design, where I showed you that there was a blocking between the ferrocyanide and the electrode surface. So this voltammogram shows it shows how the concentrations, as you increase the concentrations, the coating will interfere with the ability for the ferrocyanide to access the aptamer bound to the gold nanoparticle for a charge transfer exchange. So to wrap up this presentation, or this poster, in a sense, this presentation shows the fabrication and the modification of screen printed carbon electrodes with the modification using gold nanoparticles and selective aptamers for targeting coating. This device was tested using various concentrations of coating in sweat. And we can see that as the aptamer is bound to the surface and capturing coating, different concentrates of coating, oxidation and reduction of the ferrocyanide becomes inhibited due to the process of the conformational change once the aptamer begins to bind with the coating and captures and binds with the coating. So it blocks the ferrocyanide access to the electrode surface. So in other words, this disposable apta sensor it can be greatly used for fieldable tests, but we're still working on it. It's currently under development for the use hopefully with sweat patches to permit non-invasive determination of opiates abuse. And it's inexpensive and efficient manner. Let me see if there's any questions. Oh gosh, I don't know how to do this.
For the participants, please use the group chat to answer any question or pose any questions to Rosa. And while we're waiting, I do have one, Rosa. Have you talked to Dr. Bensali about some of his printed other biometric electrodes that look like this? So it's not paper, but it might be a flexible material?
Yes, he's actually really co-working with me on this. we decided to go with paper because one of my previous projects was developing a paper-based microfluidic device for detecting biological fluids. So, which was published, and I wanted to stick with the paper because it's really disposable and you can make multiple ones at once. So whereas the ones that he's making in his lab are, so to say, to be printed onto the skin. So I wanted to try something different. I wanted to stay in the paper-based range as well. Hopefully I answered that.
Yeah, his are kind of like Band-Aids almost.
Right, right.
Do we have any questions from our participants? Please, again, use the group chat. One other one, have you thought about this for any other type of compounds other than, are you looking at anything other than opioids?
I will be looking at other drugs, so to say, especially like your methadone and stuff. Well, methadone, because that's pretty much what other, like when you're trying to withdraw from opioids, you know, that's normally the go-to for people. It helps people come off of opioids. So we'll be looking at methadone and seeing if there's a reaction or interference. But we also want to look at a variety of opioids, not just codeine. This is codeine. We wanted to make sure that because we have a known aptamer that has previously been used in our lab, we just modified it. We wanted to make sure that that this aptamer can bind. So make sure that this electrode will work before we move on to other aptamers. We're also, hopefully by the end of the year, we'll have multiple opioids with multiple aptamers, because we already have other aptamers that we haven't yet published to show yet. So
very good. We have a couple of questions. The first one is, this is so cool. Forgive me if you've already mentioned this, but how inexpensive is it to manufacture the SPCEs?
So the conductive ink is pretty much the most expensive. We say like $70 for the 2 jars for both the silver and the carbon, but you can make roughly 5,000 electrodes with it. Whereas the paper, you buy big bulks of paper that's maybe $15, and you can make more than, based off the size of the electrodes that I'm creating. So I would say maybe, as a disclaimer, I haven't thought about the price yet, but based off my previous work, 7 cents to make one electrode.
That seems
between 7 and 10 cents.
That seems pretty reasonable.
Yeah.
The next question is, how much sweat do you need to have to be able to have a result?
So the amount of sweat that we use, that you can see in the cyclic voltammogram, was it was 13 microliters of sweat. Yeah, it was 13 microliters of sweat that was used to deposit just as blank sweat. So when we mixed it, we mixed 13 micrograms of sweat with the codeine. For granted, we weren't able to use actual human results yet, but we did a mixture, a drug mixture. So after mixing the sweat with different variety concentrations of codeine. I say roughly 13 microliters. Ugh. That answer your question, Mallory?
And the next one is, can the electrodes be reused in testing?
The paper-based electrodes, no. The ideal is for them to be disposable, because they're paper. Like, you don't want to reuse paper. Once paper gets wet, it's kind of no good any longer. It's not any good. So no.
I noticed you said you hadn't done any human studies on this. Is there a plan in the future to actually
There
try
is a
to do
plan
that?
to work with a rehabilitation center to collect sweat samples from drug abusers. So yes.
Do you see in the future that you might connect this to an app or something if you were using this for a continuous monitoring for people in rehab or other things?
Yes, I can see that in the future happening. I've spoken with Dr. Bronzali about this, because I know they have an app created for their bandages, and which I think will be cool, although it's disposable. So we're still trying to work out the kinks for the processing of this electrode. But we hope to be able to use it with the app for monitoring.
So
Also sorry.
no, go ahead.
Do you have any? If you guys would like to stay connected with me, you can check out my LinkedIn QR, which is on the left. And you can follow my science journey, which is using the QR code on the right. I need followers.
Any more questions? So then I have one. I don't want to monopolize the issue, but in the past we looked at some of this aptamer-based technology for doing not just sweat detection, but have you thought about this for regular drug detection for forensic or law enforcement uses?
One of our postdocs in our lab, she recently just published a paper that focuses on paper-based drug detection. And if I'm not mistaken, I think she's working on electrochemistry drug detection as well, using the codeine aptamer.
Yeah, we've got
not in just not in biological fluids, just as these drugs.
Okay, perfect. That's what I was looking for, because in the past we had seen some of these gold aptamers for cocaine detection, but they were very craft-based. They weren't as fully developed as what you're doing. So we do have a question from a second-year forensic student and wanted to know where you got your research idea.
No, Taylor, you haven't missed it. Well, I kind of mentioned it earlier, but you haven't missed it. My research, so previously I worked on using paper-based microfluidics for biological fluid detection. And that's for my master's research project. So to advance for my PhD research project, which is currently this, I wanted to incorporate the same, because I had a knowledge of microfluidics, paper-based microfluidics. I wanted to incorporate the whole microfluidic idea with electrochemistry. Electrochemistry seemed cool. I didn't grasp other project ideas that was inside of my lab. So I thought, let me try something different. And because I had a good a lot of my other colleagues or my fellowship colleagues, they were doing electrical engineering or some kind of engineering. I was like, I thought to myself, what can I incorporate that can have the electrical part that's different, but still have the forensics idea? So I started doing research, and I knew I wanted to keep on with the paper-based, like I said. So after doing that, I saw that you can have sensors. I like sensors. My paper microfluidic device was a sensor for biological fluids. So I knew I liked sensors. Let me see what kind of sensor can I create. And then after talking with my advisor, Dr. McCord, we came up with the whole, let's try electrochemistry. So that pretty much brought me to the electrochemistry. Then I knew initially that I wanted to work with drugs. I knew it before I even started grad school. So I was like, he was like, oh, well, this can also be the whole drug situation. So because I previously worked with biological fluids, and now I'm in my PhD where I'm focusing on the whole drug, I want to do the drug interest. I have a drug interest. Well, that sounds weird. I have an interest in research for drugs. So I pretty much collaborated everything. It was a jumbled idea in my brain, but it doesn't look like that on paper.
And related to forensic science, it's okay to say you've done drugs. Research.
Yes.
I have another question for you. Have you looked at anything other than the paper microfluidics? Maybe some
commercial
or some MEMS chips or anything that could potentially be made right there at Florida International University?
I have not. That's a good, interesting thing.
Is it again, it's a potential to talk to Dr. Bensali and Dr. Roman about, where you might actually be able to run multiple analytes in one microfluidic MEMS chip. So you could load all your aptamers in different channels.
MEMS chip.
Micro-engineered micro machine, more of a silica-based chip instead of a paper-based. It's the same thing. It's just they have the instrumentation and everything over in Amory to be able to build some of those chips. It might be something to compare with your paper-based. It won't be. It will probably not be as cheap as your paper-based, but it could be once you get the system in place.
Okay.
It's less labor intensive because it's a machine doing it than you.
That's true. I have to make sure all the aptamers, though, that I'm looking at are working. So after singly testing different opioids, making sure there's no interference with other opioids to get a simultaneously detection device, then I probably can move to that. That's great. I haven't thought about that, but I'll definitely reach out to Dr. Bensali about that too.
So when do you think you'll be looking at doing the potential human subjects testing with your current design?
My goal is to have the human testing by next summer. So around this time next year.
So
at the latest.
So we can look for a poster with human subjects next year for the conference.
I'm not agreeing to that, but hopefully, yes. It might not even be a poster. It might be a talk. You might be able to just see me in action.
And just to remind everybody, we're recording these poster sessions, so we now have Rosa on live saying that she may give a talk next year. So that's good.
I said maybe. It may be a talk. Clarification.
I have a new question for you. I'll read it out loud. I'm sorry I haven't asked any. Everyone asked the ones that are in my head, but I wanted to ask you if you changed the analyte that you are testing to 6-acetaminophen, would you need to change your experimental design?
I would not need to. I haven't tested the use acetaminophen, I would not need to change my experimental design as of yet, based off of literature research. I have been able to see, let's see if I can show you guys. I have been able to test other opioids though, well, other coating related opioids, as you can see here, but I used it in differential pulse voltammetry. And this showed that, so I have, for the left side, you have a targeting detection where we're able to see that there is a blockage of electron transfers as the concentration increases, as I previously said in about the cyclic voltammetry. And then for the right, we can see that I can detect various coating related opioids, but it's most selective. This sensor is most selective for coating at 25 nanograms, where you would need more opioids, more of like oxycodone and hydrocodone to get a signal remotely close to the coating. So based off of that, I have not been able to, this mouse is going crazy. Based off of the data that I just showed, I have not been able to explore yet other opioids.
We've got some new participants in the session. If you wanted to kind of go back over your poster for the folks that are new to the session, it might generate some more questions.
Did he say I answered his question though? Okay, yes, I was curious. Okay, gotcha. So welcome new participants. So my presentation is pretty much the application of screening printed electrodes for the detection of opioids in sweat. Due to the multitude of opioid overdose and overdose related deaths, we decided that we wanted to make a easy to use portable biosensing technology that is sensitive and can simultaneously detect opioids and biological fluids. Since there is little research available for portable drug detecting biosensing, my aim for this project has been to develop a screening method using applications of electrochemistry and aftersensing to detect the presence of opioids in sweat. So you can see the materials that I use for this project, which is a modified aptamer, 50 nanometer in size gold nanoparticle, conductive carbon and silver paste, chromatographic paper, as well as a wax printer. So the process for the materials, the process to create the electrode are seen in figure 1, where I used the screen printed machine, and it was used to place the electrodes onto the wax design. And then I used the potentiometer to measure all the voltammetric measurements that you will see in this presentation. For figure 2, you can see the schematic design of how my target binds to the working electrode of the screen printed electrode. I use nanoparticles, which is seen in the yellow. And then after allowing the nanoparticles to bind to the surface of the electrode, I then followed up and added a thiolated aptamer, which binds the thiolated aptamer to the gold nanoparticle on the surface electrode. Then I added coating inside of ferrocyanide, coating inside of ferrocyanide. Well, the ferrocyanide is the electroactive solution to create the electric charge. And then, which creates a conformational change. This conformational change is a switch on assay that occurs, and then after binding, the analyte of the aptamer to the aptamer. And we're able to get electrochemical sensing, electrochemical voltage change. The electrochemical sensing that occurs causes an oxidation between the ferrocyanide. So when the aptamer folds over after collecting the coating, the aptamer blocks the ferrocyanide's access to the electrode surface, and this creates a inhibition. It inhibits the access for the ferrocyanide to reach the electrode surface. So in the first table that you see up under characterization, we're able to characterize the nanoparticles by size and charge, comparing both bare gold nanoparticles with the aptamers coated gold nanoparticles. And it can be seen here in this chart under size that the aptamer coated gold nanoparticle increases when compared to the bare gold nanoparticle. The size increases when compared to the bare gold nanoparticle. The change in size demonstrates that there is an addition to DNA and shows that DNA has bound to the gold nanoparticle because of the gold and sulfur from the thiolated aptamer, strong binding energy. There's also, we can see under zeta potential, which is the charge, that the charge of the aptamer coated gold nanoparticle shows an increase in charge because the aptamer is blocking the surface charge of the gold of the nanoparticle to the electrostatic potential. In figure 3, we see SEM micrograms to show the interaction between gold nanoparticles and the aptamer coated gold nanoparticles with at 1 micron magnification. The right image shows that there are functionalized aptamers onto the gold nanoparticles. So we see there is an increase in the cluster of gold nanoparticles. In other words, the clusters get larger versus the left image where you see small clusters of single gold nanoparticles. So in figure 4, we can see it. Figure 4 shows the cyclic voltammogram of various concentrations in coating. It shows that there's a signal drop when coating increases. So the more coating that is present, it blocks the reaction binding of ferrocyanide, which means that the binding of coating interferes with the ability of ferrocyanide to access the aptamer bounded to gold and for the electron charge transfer. So this voltammogram correlates to what we see in the zeta potential and the size, as well as in the schematic design that's being shown. So in conclusion, we can see that there's the fabrication and the modification of the screen printed electrode, screen printed carbon electrode with modifications using gold nanoparticles, as well as the selective aptamers to target for coating. This test was done using various concentrations in sweat. The system was operated using potassium ferrocyanide as the bulk electroactive solution. And the aptamer that is bound to the gold nanoparticles onto the surface of the electrode was used to capture the coating and then this inhibits the oxidation and reduction of ferrocyanide due to the capturing of coating, which causes the conformational change of the aptamer. Of the aptamer after binding. So it blocks the surface of the electrode from creating the oxidation, from creating the electrostatic electron transfer, charge transfer exchange. And that'll be it. You know, if you have any questions, let me know. Make sure you stay connected with me on LinkedIn using the right QR code, and follow me and follow my science journey on Instagram using my right QR code. Thank you for attending.
If you have any questions, please put them in chat, and Rosa and I will be here for the next about 18 minutes. So please ask any questions you may have. We have another question. How quickly does the signal change when switching between concentration or blanks? Any hysteresis effects or issues?
So the data that you see are overlaps of different concentrations, where we use each concentration or each signal that you see are between, like, they're using different electrodes, so there is no switching between concentration or blanks. And we haven't looked at the hysterical effects or issues yet. I'll definitely look at that though. Make sure I incorporate that into my research findings.
We have 15 minutes left to go. If there's any more questions, please put those in the chat and give Rosa a chance to answer.
See. Yeah, we'll definitely check that out though. We want it to be a fieldable test, so yeah. We wanted to be able to use it on roadside testing.
I think we have a couple new participants. If you quickly want to go over this one last time, Rosa,
Okay.
for the folks.
Okay, no problem. Welcome, new participants. My presentation is on the application of screen printed electrodes for detection of opioids in sweat. We're looking at opioids due to the multitude of opioid overdoses and overdose-related deaths. Our goal is to create a rapid, easy to use, and portable biosensing technology that is sensitive and can simultaneously detect opioids and biological fluids. Since there's little research available, we want to be able to use a portable biosensing application that incorporates electrochemistry and aptosensing for detecting opioids in sweat. So it's non-invasive. You see the materials that are used in this project, and in figure 1, you can see that we have the screen printed machine to create the electrodes, and then the electrodes were printed. They were used on a wax design. They were printed onto a wax design and measured with a potential meter. Okay, sorry guys. So in figure 2, you can also see the schematic design of how my targeted analyte binds to the working electrode of the screen printed electrode, which consists of gold nanoparticles bounded with a thiolated aptamer onto the surface of the working electrode. So in the presence of coating inside of sweat and using ferrocyanide as the electrostatic solution, we're able to see a conformational change on the aptamer, on the electrode surface, where the aptamer begins to fold, blocking the access for the ferrocyanide to reach the electrode surface. And in the bottom table, we were able to characterize the nanoparticles by size and charge. And you can see that the size of the aptamer-coated gold nanoparticle increases. And this increase demonstrates the addition of DNA that has become bound to the gold nanoparticle. And then you can also see that there is a difference between the charges of the gold nanoparticle and the aptamer-coated gold nanoparticle, which The aptamer coated gold nanoparticle shows an increase in the charge because the aptamer is blocking the surface charge of the gold nanoparticle to the electrostatic potential. And the bottom figure up on the table, which is figure 3, we can see SEM micrograms to show an interaction between the gold nanoparticle on the electrode surface and the aptamer coated gold nanoparticle on an electrode surface at 1 microgram. The right image shows that the aptamers are functionalized on gold nanoparticles, and the size gets larger. As we can see, there is an enhance in clusters. And in figure 4, which are a part of my results, we see a cyclic voltogram of various concentration of codeine, where it shows a signal drop as codeine increases. So the more codeine that is present, it blocks the reaction binding of ferrocyanide. In other words, the binding of codeine interferes with the ability of ferrocyanide to access the aptamers bound to the gold electrode, bound to the gold nanoparticle for electron charge transfer exchange. So in conclusion, this presentation shows the fabrication and modification of the screen printed carbon electrode using gold nanoparticles and selective aptamers. The system operates using potassium ferrocyanide as its bulk electroactive solution. And we were able to test this device using various concentrations of codeine that was in sweat.
Any additional questions for Rosa? We are nearing our last 5 minutes of the hour. So please put those in chat so that you can get your questions answered. Or I believe it's any ideas or concepts you're looking for as well, Rosa, is that correct?
Yes. Any ideas, suggestions that I can think for future parts of this project? That'll be great.
Since you have a couple new people, do you want to tell them about the QR codes and what you're looking for there?
Oh, yes. In the left-hand corner, you can follow me on LinkedIn, or we can connect on LinkedIn. We can stay connected on LinkedIn using the left QR code. And you can also follow me in my science journey using the QR code on the right on Instagram.
Rosa's done a wonderful job of explaining the poster 4 times during this session. Fielded all the questions you see there, Dr. McCord, so I think you can be very proud of her work. All right, everyone, we're at 12:56 now, 4 minutes until the main symposium starts back up. I think I'd like to close out the poster session and say thank you again to Rosa for all your research, and I am looking forward to future presentations or posters from you.
Thank you all for attending.
Thank you all
for hosting, Kevin. And I look forward to future communication with you too.
Thank you so much, Rosa.